Hi all, welcome to Physio Exam Buddy. This video is on the neuromuscular junction or NMJ. Most commonly asked question from this topic is describe the structure of neuromuscular junction and transmission of impulses. We all know that the skeletal muscle fibers, they are innervated by large myelinated nerve fibers, and the junction between this nerve ending and the muscle fiber near its midpoint, it is known as the neuromuscular junction, which is also known as myoneural junction. And one neuromuscular junction will be present per muscle fiber except 2%. First, we will see the structure of the neuromuscular junction. Neuromuscular junction consists of a nerve as well as the muscle junction. And in case of nerve, it is the terminal buttons or the end feet of the axon. You all know that the axon supplying the muscle fiber near its termination, it loses its myelin sheath and it divides into terminal buttons or axon end feet. And you can see here that there are small numerous clear vesicles filled with the neurotransmitter, which is the acetylcholine in case of neuromuscular junction. And there are also numerous mitochondria which will supply the ATP, which is necessary for the synthesis of this excitatory neurotransmitter, that is the acetylcholine. Please keep in mind that here in neuromuscular junction, the neurotransmitter is acetylcholine and it is an example for excitatory neurotransmitter. And this is a slightly expanded view of the neuromuscular junction, and this is the neural end feet and this is the muscle part. And you can see here that in the neuromuscular junction, at the axonal terminal buttons or in the end feet, there is a blue colored structure which is known as the dense bars, and on each side of the dense bars you can see voltage gated calcium channels, and this zone which is adjacent to the dense bars, it is known as the active zone, and it is through this release site the acetylcholine will be released from these vesicles into the gap between the nerve as well as the muscle. And the space between the neuron as well as the muscle, it is known as the synaptic space, also known as the synaptic cleft. And the width of this space is usually 20 to 30 nanometer, and they contain an enzyme which is known as the acetylcholinesterase. Regarding the function of this acetylcholinesterase, it usually destroys our neurotransmitter, that is the acetylcholine, few milliseconds after the release of the acetylcholine from the vesicle into the synaptic space. And the muscle end, it is known as the motor end plate, that is the thickened portion of the muscle fiber membrane at this neuromuscular junction. And you can see here that this thickened motor end plate, it is actually invaginated to form a structure which is known as a synaptic gutter or the tuft. And the bottom of the gutter, they have numerous small folds which is known as the subneural cleft. So this is the synaptic gutter or tuft, and this is the subneural cleft, which will increase the surface area for the action of the neurotransmitter. And on the top of the or mouth of the subneural cleft, we have numerous acetylcholine receptors also present in the motor end plate. A few words about the acetylcholine receptors. This is the structure of the acetylcholine receptor. They are mainly gated ion channels having a diameter of 0.65 nanometer, and it is a protein complex having 5 subunits. And the fetal receptor complex, it contains 2 alpha and 1 each of beta, delta as well as gamma subunits. Whereas in case of adults, the gamma subunit in the fetus, it will be replaced by the epsilon subunit. In an inactive state, usually the gate, that is is usually closed, that is the channel remains constricted in an inactive state. But whenever the acetylcholine come and bind to the alpha subunit of this acetylcholine receptor, it causes the opening of the channel. And through this channel, it will allow only positive ions like sodium, potassium and calcium to move easily through the channel. It won't allow the passage of the negative ions, especially the chloride ions, because of the negative charge which is present in the mouth of the channel, which will repel the negative charge. So this opening of the acetylcholine receptor will allow more and more sodium ions than any other ions. Regarding the transmission of impulses across the neuromuscular junction, The Golgi apparatus in the cell body of a motor neuron, it will produce these small vesicles, and this small vesicle, it is transmitted through the axoplasm into the axon terminus, and in the axon terminus, acetylcholine is synthesized in the terminal cytoplasm, and this acetylcholine, it is transported into these clear vesicles, and each vesicle contains around 10,000 molecules of acetylcholine. And whenever the action potential reaches this nerve terminal, it causes the opening of this voltage gated calcium channel. Then the calcium will diffuse from this synaptic space into the interior of the nerve terminal. That is, calcium influx will occur into the neuronal end feet. This will activate an enzyme which is known as calcium calmodulin dependent protein kinase. This will phosphorylate certain synapsin proteins which will help to anchor the vesicles to the nerve terminal, and acetylcholine will move to this active zone of the nerve terminal, and it will dock at this release sites fusing with the neuronal membrane. Finally, the acetylcholine is emptied into the synaptic space by the process of exocytosis, and the exocytosed acetylcholine, this blue color one, will bind to the alpha subunit of the acetylcholine receptor, causing the opening of the ion channels, resulting in the influx of sodium ion into the motor end plate or the muscle fiber. This will increase the positive charge inside the muscle fiber, which will create a local positive potential change inside the muscle fiber membrane, resulting in the formation of the end plate potential or EPP, which causes more and more opening of the sodium channel, which will initiate an action potential, finally causing the skeletal muscle contraction. And the rapid removal of the acetylcholine by the enzyme which is present in the synaptic space, which is acetylcholine esterase, it will prevent the continued muscle re-excitation. That is, the acetylcholine esterase will break our acetylcholine into acetate and choline, and this choline part, it will be again reabsorbed actively into the nerve terminal to form new acetylcholine molecules. After each action potential, a coated pit will appear near the nerve terminal, and a protein which is named as clathrin. They are the contractile protein which coats in the area of the fused vesicle, and this protein contract and causes the pits to break into the interior of the membrane, forming new and new vesicles inside the end feet, and the acetylcholine is then transported into these clear vesicles, and finally the process is repeated. That's about the structure as well as transmission of the neuromuscular junction. Thank you.